Welcome to this episode of Mentors at Your Benchside, the podcast giving you advice, tips, and tools for getting the most out of your research. I'm Thomas Warwick, and today I'll provide some top tips to optimize nuclei extraction and counting. Single cell sequencing is a cutting edge technique for analyzing nucleic acids within individual cells from a single tissue source, enabling a granular view of their genetic and epigenetic landscapes within populations of cells. Among its key applications, single-cell RNA-seq can describe transcriptomic profiles and gene expression dynamics, while ATAC-seq addresses the accessibility of chromatin, providing insights into regulatory mechanisms. High-quality nuclei extraction is pivotal for these applications, as the purity and integrity of nuclei directly influence the accuracy and depth of the sequencing data. In this episode, I'll provide practical tips to prepare high-quality nuclei extracts and tailor your extraction protocol to your sample. Explain methods to count nuclei and assess their quality, including the pros and cons, and demonstrate how automated nuclei counting can improve your workflow. Challenges of extracting and counting nuclei for single-cell sequencing. Nuclei extraction involves removing the cell membrane via lysis while keeping the nuclear membrane intact. The resulting debris and exposed, structurally delicate nuclei pose significant practical challenges, including sample heterogeneity, sample integrity, sample contamination, nuclei aggregation, and quantification accuracy. Furthermore, these technical challenges can impact the reliability and scalability of your nuclei extraction. Tailoring your extraction protocol to your specific tissue type will help you overcome these. Keep a detailed record of all the different approaches you try to learn what works best for your sample. However, the following practical actions will improve any extraction protocol. Prepare all your buffers, dilutions, and equipment beforehand. Pre-equilibrate them to the temperature outlined in your extraction protocol. Understand what cleanup your sample will require. Keep your samples cold to minimize osmotic lysis of the extracted nuclei. Work quickly and efficiently to further minimize nuclei damage. And section large tissue samples into smaller pieces. If you have a large tissue section, consider cutting it up before you grind it to help increase the efficiency of your nuclei extraction. To ensure the heterogeneity of your nuclei extracts, eliminate as much tissue containing unwanted cells as possible. Use an automated tissue dissociator for consistent extraction. Grind your samples up using a tissue dissociator rather than manually to help improve your batch-to-batch consistency in your lab and ensure that all your samples are processed the same way. Be gentle to avoid osmotic lysis and mechanical nuclei damage. The cell membrane protects the nuclei in regular healthy cells. However, when you lyse cells for nuclei extraction, the nuclei become exposed to everything previously kept outside of the cell. Because the nuclear membrane is more fragile than the cell membrane, the nuclei are susceptible to osmotic lysis. Working quickly and keeping everything cool minimizes this. They are also extremely fragile and susceptible to mechanical damage, so perform all manual handling of your extracted nuclei gently. Optimize filtration to eliminate sample debris. To eliminate as much cellular debris from your nuclei sample as possible before moving to sequencing, aim to learn what specific filtration your sample will require. The size and abundance of cellular debris will vary depending on your tissue type and lysis method, so choose the pore size of your filter accordingly. We will discuss other steps to eliminate sample debris later in this episode. Economize your sample when performing quality control checks. Because nuclei extraction protocols often yield small sample volumes, which may have only a sparse number of nuclei, aim to minimize the amount you use for nuclei counting and quality control to ensure you have plenty left for sequencing. Use fluorescence rather than bright field methods to count your nuclei. Although your choice of using fluorescence rather than bright field methods to count nuclei may be constrained by the instruments you have, fluorescence-based methods offer significant advantages. Bright field nuclei counting methods using trypan blue. Trypan blue is an azo dye that binds to and stains intracellular proteins, and it remains a popular choice to test cell viability. Due largely to its size, 873 Daltons, it cannot pass through the cell membrane and therefore stains dead cells, cells with compromised membranes, isolated nuclei, and cell debris. Under the microscope, live cells will be clear, that is, unstained, while dead and degraded cells, nuclei, and cell debris will be blue/blue-black. To prevent the quality of your trypan blue from complicating your nuclei counting, inspect a few microliters under a microscope. If you see some crystallites or contaminants in your trypan blue solution, try centrifuging it at a relatively high speed. Isolate the top portion of the solution, filter it, and re-inspect it to see if it's any cleaner. Another option is to run the trypan blue through a 0.2 micrometer filter to remove the large debris. If neither method works, to remove the debris, buy new stocks. Gently mix your sample and the trypan blue at a 1:1 ratio in a fresh tube and incubate the mixture for 3 to 5 minutes. Then proceed to nuclei counting. For a detailed explanation of testing cell viability, see reference 4 in the corresponding online article. Fluorescence nuclei counting methods using acridine orange and propedium iodide. Acridine orange and propedium iodide, or AOPI, are fluorescent dyes that intercalate nucleic acid. Acridine orange is the smaller of the two, at 265 Daltons, and it can cross the membrane of living cells. And it fluoresces green at 520 nanometers. Propedium iodide is larger, at 668 Daltons, and is excluded from living cells like trypan blue. It can, however, cross the compromised membranes of dead or dying cells, and it fluoresces red at 618 nanometers. So, any successfully extracted nuclei will fluoresce red, and any unlysed cells will fluoresce green. This means you get AO and PI in any dead cell/nuclei. However, the green fluorescence from AO is transferred via Foerster resonance energy to PI and it is quenched. This results in a binary situation where nuclei are red and intact cells are green. Because AO and PI bind to nucleic acid, any object in your sample that does not have a nucleus, such as debris or red blood cells, won't fluoresce. Check out the corresponding online article to see what cells stained with AOPI look like. Compared to trypan blue, using AOPI provides more accurate estimates of the total number of cells and cell viability, and the results are more objective. Because AOPI are fluorescent dyes, they have superior detection sensitivity than trypan blue. Although it's best to pre-cool your other reagents, leave the AOPI at room temperature to avoid temperature-induced fluorescence quenching. The optimal ratio of AOPI to sample is 1:1. Add the two to a fresh tube and gently pipette up and down a few times to mix them thoroughly, but remember the nuclei are extremely fragile. Avoid vortexing if you can. Note also that AOPI does not require an incubation time, so you can proceed straight to nuclei counting after mixing. Perform pre-sequencing quality control checks on nuclei samples. Many downstream applications for extracted nuclei are expensive and lengthy. Plus, you get a large amount of complex data, which will be either degraded in quality or hard to interpret if the starting sample is poor. Therefore, spending some time on quality control to gauge the suitability of your sample for your intended application is paramount. Visualize and quantitate debris. Debris left over from the nuclei extraction can negatively impact downstream results, such as sequencing data, especially if there's a significant amount of it. Debris can also clog microfluidic devices and take up wells intended for nuclei, leading to wasted time and resources. Therefore, it's crucial to assess the amount of debris in your sample before continuing to ensure it's fit for purpose. Check out Figure 2 in the corresponding online article to see when the level of debris in your sample is fine and when it's likely to cause problems in your downstream applications. If your sample contains lots of debris, all is not lost. You can purify it using the anti-nucleus microbeads from Miltenyi Biotech. which specifically bind to the extracted nuclei to eliminate unwanted debris and live cells without impacting your yield. Check out the corresponding online article for a link to this product. Measure extraction efficiency. Calculating the extraction efficiency of your nuclei lets you assess the effectiveness of your nuclei extraction protocol. It's defined as the proportion of nuclei extracted from the cells in a given sample relative to the total number of nuclei present, either extracted or remaining unlysed in the cells. You can calculate it using these following steps. Determine the total number of cells in your starting sample. You can do this using a hemocytometer or automated cell counter. Calculate your expected nuclei count. Assuming each cell contains one nucleus, your expected nuclei count should ideally match the initial cell count. Count the number of nuclei obtained after extraction. If you've used fluorescent dyes like AOPI to label the nuclei, you can count them using fluorescence microscopy or an automated cell counter. Calculate the nuclei extraction efficiency using the following formula. 100 times nuclei per mil divided by, open brackets, nuclei plus cells per mil, close brackets. What is an acceptable nuclei yield and extraction efficiency? While low nuclei yields will impact the quality of your data you get from single cell or nuclei sequencing, there's no hard and fast rule regarding what yield to aim for. However, empirically determining your nuclei yield allows you to quantify an acceptable value suitable for your downstream application. The same applies to calculating the nuclei extraction efficiency. If there are 100 events on screen, but only 50 are nuclei, you have 50 cells that will not get sequenced. An extraction efficiency of 50% probably means you must extend the lysis period or refresh your lysis buffers and try again. While you should aim for as high extraction efficiency as possible, no target value applies to all applications. Providers of downstream applications may provide guidance for these quality metrics. Consider automated cell and nuclei counting. As mentioned, precise quantification of nuclei is critical for downstream applications. Manual nuclei quantification is slow and prone to user subjectivity. Automated nuclei quantification using a cell counter is a superior choice for almost all downstream applications because it's unbiased, standardized, scalable, fast, and accurate. Count cells without slides using CellDrop. The CellDrop automated cell counter offers all the benefits of automation listed above and lets you fine-tune the following event detection criteria: cell diameter and roundedness, nuclei diameter and roundedness, fluorescence threshold for each channel, and irregular cell types and shapes. Plus, it can count cells without disposable slides to reduce the number of consumables you need and provide clearer images. Not only does this reduce the carbon footprint of your lab, keep costs down, and help any green initiatives at your workplace, but it also offers a performance benefit. The flat, sapphire surfaces and lack of requirement for consumables minimize the CellDrop's measurement-to-measurement variability, giving you more reliable counts than instruments that require plastic slides or hemocytometers. Furthermore, precision-controlled sample arm allows you to use as little as 2.5 microliters of your nuclei extract for counting and quality control, meaning you have more left over for sequencing. It's even available in green. Visit the Genomics website to learn more about the CellDrop. From practical tweaks such as pre-chilling your reagents and working quickly, to taking the time to investigate your sample's cleanup requirements, the tips outlined in this article will help you prepare high-quality nuclei extracts. Tailoring your nuclei extraction protocol to suit your sample and investing time in quality control will ensure superior single-cell sequencing results. Frequently asked questions. How can I avoid nuclei clumping in my extracts? First, ensure that all your cells are healthy when you begin to extract nuclei from them. You can remove large clumps by filtration or density gradient centrifugation. You can also modulate the osmotic pressure of the extraction buffer by adjusting the concentration of osmolytes such as sucrose. If your nuclei can tolerate it, Balance the electrostatic interactions between nuclei by adding or adjusting the concentration of salts such as sodium and magnesium chloride. What is the accuracy range of the cell drop? The cell drop offers a flexible sample range from as few as 700 to 25 million cells per mil, depending on the sample arm height. At the default sample arm height of 100 micrometers, the accuracy range is 100,000 to 10 million cells per mil. The lower arm height of 50 micrometers allows the use of lower measurement volumes to conserve precious sample. I need to inspect my nuclei under a high-resolution microscope and count them. Can I use the microscope slides on the cell drop to reduce sample wastage? Yes. While the cell drop does not require microscope slides to count the nuclei, the sample arm can be set high enough to accommodate them, so you can use them on the cell drop to cut down on the sample volume needed for nuclei counting and analysis. So that's it for nuclei counting. Check out the episode description for links to related articles and resources. And don't forget to subscribe to the podcast to get more help and advice from mentors at your Benchside.